Good evening, Mr. CTO. Thank you for coming. This proposal is about using data science for drug design and clinical outcome prediction for personalized cancer immunotherapy. This proposal has been approved by Pfizer CEO, but we all know that without your support and approval, this proposal will not go anywhere. So for this proposal, we're asking for a hire of 8 data scientists who have immunology background for an annual budget of 0.8 million at the first phase. Half of the data scientists will focus on rational drug design. The other half will work on predicting clinical trial response from patients. This proposal is based on the following facts. First, Pfizer is eager to become an impactful player in cancer immunotherapy. This cancer immunotherapy is a new, rapid, expanding oncology branch for cancer treatment so far. The current cancer immunotherapy drug design lacks a rational approach. It is still based on experience and traditional trial and error testing. We all know that deep learning is a powerful tool to capture non-linearity. In immunotherapy drug design, molecular interactions are highly context-dependent and non-linear. So our hypothesis is that we can use deep learning to do a rational drug design. Deep learning has huge potential in predicting clinical response from patients taking certain drugs, and ultimately, we hope that it can enable us to prescribe personalized immunotherapeutic medicine for any given patient. Before going deep the detail about this proposal, I'd like to share with you a little bit of background about cancer and the immune system. So we all know that cancer is caused in part by mutation in the DNA sequence. So with that mutated DNA sequence, it's going to generate mutated proteins in the cancer cells. An adaptive immune system will present those mutated foreign proteins in cancer cells on the cell surface. Normally, the T cell is going to recognize those specific antigens and kill those cells. This is going to lead to the T cell expansion. However, in cancer cells they generate some specific signals that are going to be shut down the T cell activities. The checkpoint inhibitors are the most successful type of immune therapy for cancer treatment so far. The main function of those inhibitors is to remove the inhibitory effect from the cancer signals and to reactivate the T cells so the T cell can do their jobs and kill those cancer cells. However, sometimes if you have so many T cells activated, it can cause major side effects through autoimmune activity. So this has to be regulated carefully. And not every patient can react with immunotherapy positively because some cancers have more mutations than the others, and not all patients have active and available T cells to fight cancer cells. And there's many other factors we don't know yet. The first part of this proposal is to predict immunogenicity of antigens. So this will have 2 tasks for this proposal part 1. The first task is to predict the interaction between MHC and antigen complex on the cell surface. So we all know that genetic mutations from DNA are going to cause the mutation of proteins. And those proteins are going to eventually be degraded by cells into multiple small fragments. Some of the fragments will be presented on the cell surface by MHC, but some of them don't. So we have to predict, use machine learning to predict what peptides or antigens, sometimes called antigens, and how they're going to be represented on the cell surface by MHC. The task 2 is to predict the T cell binding and expansion based on the patient T cell profile and antigens presented on the cancer cell surface. The ultimate goal is to determine which immunotherapy drugs likely to be most effective for any given patient. For MHC binding prediction, here is the protein structure for MHC on the right side. It's composed of about 200 to 300 amino acids. And the top of this complex is a peptide in green color. So those peptides very short, very small. Those are from mutated proteins from cancer cells. So there are thousands of MHC anneals in human population. Each anneal capable of binding a distinct set of peptides. Our objective is to predict whether our MHC anneal will bind a given peptide. There are a lot of public available data which are based on experimental results. That is for the interactions between specific MHC anneals and specific peptides. Here is the table, shows that information. The first column is anneal name. The second column is the peptide sequences. And the last column is the binding affinity between these two components. As you can see here, there's one row. It's the binding affinity is 5,000. That means the binding between MHC and the peptide is pretty strong. And there's another row, the binding affinity is 20. That means the binding between these two components are pretty weak. There are also other public available data regarding the interactions between T cell receptors and MHC antigen complex and the cell surface. So sometimes their interaction is negative, sometimes is positive. So we can use all those public available data, either from other researchers or from experiments. And we use those data to train our models, and we can predict new results based on the new antigens. Here, I'd like to share with you our preliminary machine learning data. Our goal is to predict what antigens are presented on the cell surface by MHC. So MHC anneal we use is HLA-A0201. The MHC peptide binding data was obtained from this website. We used cross-validation by dividing the whole data into two parts, training and test data sets, use a ratio of 9:1. So the machine learning results are going to be shown based on the test data set. Initially, there was a few researchers, they used carrots in our environment to predict the interactions. We were able to reproduce the results with TensorFlow in Python environment, and we confirmed the high prediction accuracy at 95%. This is the data from trained our model used public available data. And as you can see here, only selective peptides can be presented by MHC on the cell surface. And you can see that position number 2 and position number 9, they prefer very specific peptides. We used feed-forward neural network model, and we got a prediction accuracy at 94.8%, pretty high. And class 0 is the no binding between MHC and antigens. Class 1 is weak binding between these two components, and class 2 is strong binding between those two. And the blue colored dots represent correctly predicted events. The red dots represent incorrectly predicted events. We also tried convolutional neural network, and we got a similar accuracy at 92%. However, when we tried random forest, the accuracy dropped from 95% to 82%. So this data suggests that to model the interaction between MHC and antigens, probably it's better to use neural network. Quick summary for proposal part 1, predicting immunogenicity of antigen. Although I only show you the modeling data from model the interaction between MHC and antigen complex on the cell surface. With that excellent modeling data I just showed you, I hope I have convinced you that if we properly use the data science, we may be able to achieve our ultimate goal, that is to determine which immunotherapy drugs likely to be most effective for any given patient. The part 2 of our proposal will be presented by Anne Nugent. Thank you very much.